So this is the Smad pathway. This is significant because signal proteins of the TGF-beta superfamily act through the receptor serine threonine kinases and these SMADs. So SMADs are a family of transcription regulators. They play major roles in intracellular mediators that transduce the TGF-beta signal from the cell surface to the nucleus. There are three types of SMADs: R-SMADs, which are the receptor regulated, as shown in blue in the figure, and these are SMADs 2 or 3. Co-SMADs shown in brown, which are SMAD4s, and then I-SMADs, which are inhibiting SMADs. These are not shown in the figure, but I will talk about those in a bit. And then TGF-beta proteins, these are not SMADs. These are transforming growth factor betas, and these act as the signaling ligands. So the TGF-beta proteins act as dimers. So once they are dimerized, they can bind and bring together the type 1 and type 2 receptors, where these receptors are the serine threonine kinases. And so once a ligand is bound to the type 2 receptor, it will recruit the type 1 receptor and activate the type 1 receptor by phosphorylation. The type 1 receptor will then go on to phosphorylate its target protein, which is in blue, the SMAD 2 or 3. This SMAD has two domains, one being the SMAD and then the other being a DNA binding domain. The DNA binding domain has a nuclear localization signal, which is hidden within the two-part domain, which is why the SMAD must be phosphorylated by the type 1 receptor so that NLS can be exposed from the conformational change that occurs once that SMAD is phosphorylated. So once the R-SMAD is phosphorylated, it is oligomerized by a co-SMAD and also another R-SMAD. Together, the complex translocates into the nucleus, where it can then collaborate with other transcription regulators to control transcription of particular target genes, usually have to do with growth and development. So negative feedback is what regulates this pathway, along with the target activated genes activated by the SMAD complex. There are also inhibitory SMAD genes that get activated with the target genes, where these I-SMADs bind to the cytosolic tail of the activated receptor and inhibit its signaling ability in at least three ways. So one of these being it competes with R-SMADs for binding sites on the receptor. So that decreases the R-SMAD phosphorylation. It recruits a ubiquitin ligase called a SMRF, which just stands for SMAD ubiquitination regulatory factor. So that ubiquitinates the receptor, basically makes it so that the receptor is tagged to be degraded and then therefore can no longer be used. It also recruits a protein phosphatase that dephosphorylates and inactivates the receptor. The inhibitory SMADs also bind to the co-SMAD, which then inhibits it by preventing binding to the R-SMADs or by promoting its ubiquitination and degradation. SMADs will shuttle between the cytoplasm and the nucleus continuously, where they are dephosphorylated in the nucleus and exported to the cytoplasm. Then once they are in the cytoplasm, they are rephosphorylated by active receptors and will stay in the cytoplasm until another TGF-beta dimer ligand comes in and turns that signaling pathway back on, which down the line will phosphorylate that SMAD. This is a more realistic figure of what it looks like for these SMAD pathways. So as you can see, there is a lot of overlap. Obviously, there is not just one signaling pathway that is being ran inside of our cells, inside the body. Even this figure, I'm sure, and you guys know that this is probably still very, very oversimplified, but it does give a better visual of what it looks like as far as what is actually happening around it.